can see the presentation.
Yes, we see it. It's okay. So, well, I think that we can begin. And well, there is some noise there. I don't understand what is happening. Okay. Well, it's a pleasure for me to present our today's speaker, Aleksey Belyaev from Moscow State University. And Aleksey will continue the cycle of presentations about blood coagulation, platelets, and all the things. Very interesting topic. So please, Aleksey, you can start.
Vitaly, thank you for invitation to participate in the seminar. And today I would like to give a talk about some biomechanical features of the initial stages of platelet hemostasis. And of course, in the very beginning, I would like to introduce you to this phenomenon and mostly to the most important parts of the biophysics of thrombosis and hemostasis. Well, the hemostatic reaction is a phenomenon in our organism that is meant to prevent bleeding. Whenever there is a trauma or a rupture of a vessel wall, the blood starts leaking out, and to prevent this bleeding, there are several mechanisms in our bodies that are meant to cover this leakage. First of all, whenever the injury happens, the platelets get triggered by the subendothelial collagen or some other molecules that they are adhesive to, and they start adhesion and aggregation at site of the trauma. Secondly, the platelets can go through a biological process which is known as activation, which shifts their mechanical, biochemical properties drastically. And after this activation, platelets are capable of signaling. They can communicate to each other. They can also communicate to blood plasma and whatever components are there involved in hemostasis in the blood plasma. And after that, after this happens, of course, there's a second branch of hemostasis and thrombosis, which is called plasma coagulation. The final product of this process is, of course, the fibrin gel, which should be formed. However, the flow and drag forces from the flow inhibit this process. And platelets need to scaffold out all the hydrodynamic effects, and fibrin gel can be formed in between those platelets within the aggregate. And of course, there's always a place for different biochemical regulation due to several proteins and smaller molecules that are present in blood plasma. All these branches, both of these branches of hemostasis are intertwined completely because the adhesion and activation relies on several proteins that are present in plasma. And at the same time, platelets are necessary to form fibrin gel, and they participate in biochemical regulation. And the regulation of platelet properties is, of course, mediated by the plasma proteins. So when the hemostasis fails...
Aleksey, yeah. I'm sorry. Looks like we have a question from the audience. So yeah. Please, Aleksey, who is guest, there is no written name here. Do you have a question? No. Well, I don't. Well, it was a hand raised, but it disappeared. Looks like question disappeared. So please continue. Sorry. Yes.
Sorry. This is no. This is wrong. This was wrong. No. I haven't question. No. Sorry.
Okay. So no question. Yes, because when you say this is wrong, I thought you were going to say that everything about these platelets and all these things they were wrong. I was kind of a little bit scared, but okay, apparently it's okay. So Aleksey, you can continue, please.
Okay, thank you. I hope this is not completely wrong. Some parts of it are actually supported by known publications and so on. So here you can see the experimental photograph of the small thrombus formed as a result of the trauma. I think it's laser-induced injury in microvessel of a mouse. So the flow is quite intense here, and the platelets play a significant role in hemostatic reaction in those platelets where the drag forces on the wall of the vessel are quite high, namely arteries, arterioles, and venules. For veins, this is probably not very important for platelets to aggregate, and the plasma coagulation can take place at the site of the injury without involving platelets, but then some more complex things can happen, both including red blood cells and platelets also, so it's quite complicated to tell. I will focus today on arterial and microvascular thrombosis and hemostasis. So when the hemostatic reaction fails, both by being unable to form platelet aggregates or by over-aggregating those platelets or by failing to coagulate the plasma and form fibrin gel, there can happen some bad consequences from that, including thrombosis or either complete vessel occlusion or embolization of several parts of thrombi and so on. Or adversely, hemorrhaging is also a bad consequence of failure of the thrombotic and hemostatic response. When we look closer at platelet aggregates, we can see that firstly, these aggregates are quite dense, and secondly, we can see that several morphological changes happen to the platelets after they aggregate, especially when there is a flow in the flow chamber, for example, as in these experiments, or in the blood vessel. Normally, the platelets circulate in blood in the form of these spheroidal oblate plates. That's why they call platelets, and they can bind several proteins. I will tell a bit later about that. But mostly they are inert and they cannot bind to healthy endothelium. When they are aggregated here or adherent to some substrate, for example, some proteins that are present, they turn into this activated state. And the difference between non-activated and activated platelets is quite obvious from the scanning electron microscopy. And first of all, we notice the shape change. They are no longer oblate spheroidal in this activated state. They also have some protrusions from their outer membrane, and they are more likely to be spherical, but with these irregularities in their shape. They get softer also, and they bind to each other stronger than in the non-activated state. But normally they circulate in this form, and you can see here in this picture I very like that there are some small bulbs or like dots on the surfaces of these platelets. Apparently there are some proteins absorbed, or basically it is not clear which proteins these are, but there are a lot of smaller proteins in the membrane, incorporated in the membranes of those cells, and mostly they are adhesive proteins or receptors that allow platelets to probe the media around them and probe not only chemically but also mechanically. So the motivation for this particular study was actually a recent pandemic of COVID-19, and there's a lot of publications about how microvascular thrombosis is one of the most popular consequences of infection of SARS-CoV-2. Well, normally, as I have mentioned, the endothelium, the endothelial cells that reside at the inner surface of the vessel, they have anticoagulant and antithrombotic properties. However, if some stimulus is being transferred to those cells, they can get activated, and this activation of endothelial cells usually causes secretion of several proteins into the bloodstream. One of the most important proteins and abundant proteins that is being secreted to the bloodstream is von Willebrand factor. Those long blue strings represented in this cartoon here are actually the released von Willebrand factor, and you can see that upon this release, these long molecules are attached by one of its ends to the endothelial cell, and the rest of the molecule just flows along with the blood. So these proteins are very sticky, especially to blood platelets. There are several factors that can trigger this endothelial activation. It can be hemodynamic changes, it could be mechanical impact or bending causing some trauma or rupture happening to these cells. And it can also be some biological trigger, for example, virus infection or biochemical stimulation, for example, by cytokines here, and so on. So these sticky von Willebrand factor molecules are quite important in this situation because they can bind a lot of platelets, and those platelets can get activated, and those platelets can potentially cause the thrombosis, the formation of microthrombi, both red, containing red blood cells, and purely white or containing only platelets and von Willebrand factor. And also these microaggregates may attract some leukocytes, for example, neutrophils and monocytes as well. So this is very intriguing part of hemostatic response system to investigate. Upon the activation, the secretion of von Willebrand factor happens, and it is very intriguing process itself. Of special capsules or granules from the endothelial cells. Those capsules are called viable Pallade bodies, and then they contain mainly von Willebrand factor in a long multimeric form, which is a linear concatemer of this protein. So it's a linear protein, a string, but normally it is in a coiled conformation. Inside those viable Pallade bodies, they're actually very compact, and they have a shape of spirals densely packed to each other. But when the exocytosis happens, the string is being unwinded to the blood flow. After that, after this von Willebrand factor is exposed to the blood flow, the shear forces from the blood flow tend to stretch, uncoil this protein, and make it active and available for platelets to attach to it. So that's the basic mechanism that every hemostasiologist has in mind when it speaks about the endothelial activation and von Willebrand factor mediated platelet aggregation. Besides that protein, there are several ways for platelets to aggregate. So the adhesion of platelets mediated by von Willebrand factor happens without any additional condition. So only the presence of this protein is required, and the ability of special receptors, GP1B/5/9 complexes on the membranes of platelets is required. So this receptor and this long multimeric protein works as at the very first stages of platelet aggregation. However, besides them, there are several other receptors. For example, there are receptors for collagen. There is a receptor that allows platelets to bind fibrinogen, either adsorbed fibrinogen or fibrinogen from the plasma, and this one is also very important. Here it is called GP2B3A, but mostly it is known by the name as integrin alpha-2B beta-3. And in many publications it's called this way, but those namings are just synonyms. The difference between those receptor and ligand families are so that GP1B works always. It is always active. It is always ready to bind von Willebrand factor. It binds quite fast, but it's relatively weak. However, those integrins, alpha-2B beta-3 or GP2B3A, or just integrins, are very strong, and they are absolutely required to form greater platelet aggregates or the whole thrombi, but they are quite slow. Firstly, they're slow because they require some stimulus to get activated. To get sticky, they need the calcium level inside the platelets to be raised up. This level of calcium can be raised by several mechanisms, either by biochemical stimuli or by binding those receptors with collagen, or by stimuli coming from GP1B receptor when it binds to von Willebrand factor. Anyway, when the calcium oscillation happens inside the cytosol of platelets, those GP2B3A integrins get activated and then get sticky. Also, I would like to draw your attention to this nice picture of electronic microphotograph. I think it's fluorescent microscopy, showing with different colors, different components of hemostasis system at the very beginning of the platelet aggregation. The magenta color represents those ultra-long von Willebrand factor strings, and the green color here represents the platelets adherent to these von Willebrand factor strings. And the blue color represents the nuclei of monocytes, which are apparently being attracted by those platelets that bind here. And it is known that monocytes can contribute to atherosclerosis. So probably the adhesion of platelets to von Willebrand factor is not only the problem for hemostatic reaction, but it can be also one of the triggers of development of atherosclerotic plaques in arteries and microvessels probably as well. So yeah, this is a very important and intriguing question. The question is, how can we study it? How can we investigate what is going on there with the power of mathematics and computational sciences? So the main goal here is, of course, biophysical, to identify the biomechanical factors that control the dynamics of platelet aggregation on the inflamed endothelium of microvessels. But there are several problems to resolve. First of all, we need to improve the existing model to make it accounting for mechanical activation of platelets. Then we need to adjust the parameters of this model to make it relevant to realistic situations. And then we should study the possibility of mechanical activation of platelets in this model within realistic or physiologically relevant hemodynamic conditions. As the main method of this particular work, the computer simulation technique was chosen. We use three-dimensional computer model, and there is a snapshot of what is going on in this system. Generally, we have three components, three main components, say, like red blood cells, platelets, and von Willebrand factor multimers. Everything rest, it can be added, of course, if needed. For example, we can add extra proteins here to diffuse and somehow interact with the cell membranes. We can add extra cells like monocytes, for example, here to roll inside the vessel and see what's happening. But for this setup, we choose only these three objects. We simulate, of course, the blood plasma, but we have chosen the continuum approach for that. However, we use the realistic erythrocyte mechanics, and we represent the surface of erythrocytes as the mesh of interconnected nodes connected by elastic elements. The same model we use to represent blood platelets, and we represent inactive platelets as oblate spheroids. And also we account explicitly for the von Willebrand factor. We allow this protein to change its conformation depending on hemodynamic conditions, and we allow this protein to get activated, of course. On this slide, you can see those three types of objects, and you can see the particular mathematics that stands behind each component of the model. For example, for fluid mechanics, the lattice-Boltzmann approach is used. It's quite a powerful tool when it comes to computer simulations because it gives a realistic blood flow, viscous fluid flow solution rather fast. It's easy to implement, especially on MPI architecture. And the only modification that we use in this particular work to existing classical lattice-Boltzmann approaches is that we use a fluctuating lattice-Boltzmann approach. There is a fluctuating term which is meant to describe thermal motion of the fluid. This is needed to give these von Willebrand factor proteins more realistic dynamics, because apparently the thermal noise has an impact on these proteins. Then for description of movement of membranes of all the cells that are present in this system, we use particle dynamics simulation, which is quite a usual approach. So we rely on Newton's law of motion. And of course, we use the coupling between those two parts, between fluid dynamics and particle dynamics. And force, which couples the particle to the fluid, is similar to what is described by the Stokes law. So there is a difference of velocities between the velocity of the particle and the velocity of the fluid, and there is a constant coefficient which relates the drag force and the velocity difference. I suppose I should skip all the details, but I would only like to draw your attention that in the current version of the model, we're using a different constitutive law for the stretching force. Usually there is some neo-Hookean power law used here, but we instead decided to use finitely extensible non-linear elastic, or FENE, approach in this version of the model to make those red blood cells deform more realistic in the conditions of elevated shear stress. Also, this law is used for modeling the bonds between those beads representing parts of von Willebrand factor. And here I would like to tell about the new feature that hasn't been presented before. We intend to give an insight on the mechanical activation of blood platelets after the immediate adhesion to von Willebrand factor. So we have modeled several receptors on the membranes of those cells. On each cell there are about 100 receptors of GP1B kind, and also there is a considerable amount of integrin receptors, which are normally reside in inactive state. So they're passive, they cannot bind to anything. However, if the force is being applied to GP1B receptor for a considerable amount of time, there is a probability, and these values are taken from the experimental work by Zhang et al. There is a probability that a platelet gets activated from this mechanical signal. This signal can be transformed through the membrane by the special structure of the protein, of GP1B protein is a transmembrane protein. There is a part that can be uncoiled, and it is called mechanosensitive domain. And it is not quite well known what's happening inside the platelets after this uncoiling happens, but it is known that it can cause the rise of intracellular calcium level, and this calcium always activates the GP2B3 integrins. So we use this phenomenology to model the activation of blood platelets by mechanical forces. We also apply several non-bonded interaction to our system, of course, to first of all to imitate hydrophobic solvent effect for von Willebrand factor monomers. This is basically meant to reproduce the well-known feature of this protein to take the compact conformation when there is no flow, and to extend when there is a shear force exerted on this protein. We also use wall repulsion potential like this. It's non-bonded also, of course. And the interaction, the adhesive interaction between the platelet receptors and von Willebrand factor are modeled by this non-bonded potential, which is Morse potential actually. So it basically behaves as harmonic potential near the minimal distance, near the equilibrium location, but it has non-linear shape, and it is a breakable potential. So if you stretch the bond far enough, then the energy will decay and the force resulting from this potential will also fall down to zero. Next up, the red blood cell mechanics has been validated by comparison with existing experimental data. For example, this is an example when we reproduced the optical tweezer experiment. This is quite a benchmark for hemodynamics. Here we stretch the red blood cell and we measure the distance between its ends in longitudinal and transversal direction, and we see that it reproduces the non-linear response observed in the experiments. Next, we can independently tune not only mechanical stiffness coefficients, but also the coupling coefficient by comparing these tongue threading dynamics of the cells, comparing to the experiments by these authors here. And we measure the frequency of this tongue threading and relate it to the shear rate. And we see that with our parameter choice, mainly the coupling coefficient, we get a satisfactory coincidence of simulation with the experimental results. Next up, the platelet mechanics and the drag forces was validated also. It is very important that we measure the drag force on the platelet correctly, and that this value coincides at least with theoretical estimations of what's going to happen. So we here measured the force and the torque on a platelet depending on the shear rate, and compared to the well-known Goldman-Cox-Brenner analytical solution, which is represented here by this dashed line. And this solution was obtained analytically in the case of vanishing distance between a platelet and a surface, a plane wall. So we see that if we decrease the distance between a platelet and a wall, here in our simulations, we get closer to this theoretical estimation here. So the triangle is the ideal situation for this comparison. So therefore, we were able to independently tune this coupling coefficient or drag force coefficient for each particular object in our model, independently for red blood cells, for platelets, and for the protein. Then also we checked if the platelet motion in the shear flow happens correctly, and the well-known result from that is the Jeffrey orbits. Jeffrey orbits when this oblate spheroid just jumps and tumbles along the path of its traveling here, and we were able to reproduce with our simulations this type of behavior. Finally, also, well, we performed some validation for von Willebrand factor. It can uncoil if the shear rate exceeds some known threshold. This threshold is very close to what we observe in the experiment, so I will not tell a lot about this. If you're interested, I can tell after this presentation some details. Most importantly, there are several activation processes that are known to be important for platelet hemostasis. And before this work actually happened that I'm telling you today, we validated our von Willebrand factor model in terms of its activation. So from the molecular point of view, what happens there is that there are two domains, A1 and A2, within this protein that are very close to each other, and they block each other from being active. In particular, the A2 domain just interferes with the ability of A1 domain to bind GP1B receptor of platelets. However, if we apply some pair of forces, apply some tension to this protein, then those domains separate from each other and A1 can bind platelets. Apparently, this is what happened in the experiments in flow chambers by Fu and co-authors here. And this was reproduced in our model, not just qualitatively, but also quantitatively. Now, back to the platelet adhesion. The simulation protocol for this work was as follows. First, we put some von Willebrand factor multimers of fixed length into this system. The system was represented by a rectangular box with top and bottom surfaces represented by those impenetratable and solid walls, and the other boundaries were just free to go. Periodic boundary conditions in lateral direction was applied. We grafted these proteins by one of their ends. Then we equilibrated in the thermostat, allowed those proteins to acquire their compact conformation without any flow. Then we turn on the Poiseuille flow by inducing the pressure gradient along this lateral direction in the system, and we saw how those von Willebrand factor multimers just stretch out in this flow. After that, after several milliseconds of this pressurized equilibration, we put several platelets in random positions, but at a fixed distance from the bottom wall. And after that, we turn on again the simulation. We started sampling, and after simulating 100 milliseconds of physical time, we stopped the simulation and turned to the post-processing procedures, which involve some Python scripting and visualization with the ParaView. Important question, how we verify this situation, how we tune the parameters, especially the unknown parameters, the adhesive energy of platelets to von Willebrand factor in both inactive and active states. So this was done by a comparison with existing experiments in the flow chambers by Schaff, by Kulkarni, and by Shi and their colleagues, of course. And it was possible to tune our system to represent, at least qualitatively. I think the quantitative agreement is also satisfactory. Important thing that we notice here is that initially the density of adherent platelets is quite low, but if we increase the shear rate in the system, this number of adherent platelets starts growing, both in experiments and both in our model. We attribute it to the activation of von Willebrand factor, of course. After reaching the peak value, these platelet adhesion starts decreasing rapidly due to the prevalence of direct forces on those adherent platelets. Now let me turn to the results. I hope I still have time to tell you about most important data that we acquired through this modeling. First of all, we varied the threshold force required to activate von Willebrand factor multimers, and we see that the lower force, 10 piconewtons, results in the onset of platelet adhesion at the lower values of the shear stress. So normally it happens at around 5 Pascal or a bit less than 5.3 pascals. And this is what's reported to see in the literature. However, if we shift this parameter, it has the most significant influence on the model behavior. So if we just decrease it to 10 piconewtons, the onset of platelet adhesion shifts to almost 1 Pascal, or even 0.5 pascals. When we increase this parameter, in contrast, the platelets are not likely to adhere to non-activated von Willebrand factor, and they start adhering to it only if the shear stress is as high as 10 pascals. And this behavior, the number of attached platelets, correlates, of course, with the number of active von Willebrand factor multimers present in our system. As we see here, for the same simulations, these dots represent the simulations, those lines represent the theoretical estimations If we take a look at several snapshots of the simulated system for certain hemodynamic conditions and for certain parameter values at different moments of time, we can see that interesting mechanics is happening in the system. So in the very beginning, one platelet can attach to a low number of activated von Willebrand factor in the system. It can bind there, but due to drag forces, it's being dragged along the direction of the flow here, and it activates more and more von Willebrand factor monomers. Then this amount of activated von Willebrand factor monomers also attracts other platelets to bind here, and therefore we found that it's not only von Willebrand activation that is needed for platelet binding, but also platelet binding causes further activation of von Willebrand factor. So there's probably a positive feedback loop in those initial stages of platelet adhesion, which lets those platelets aggregate and adhere in the places where other platelets are already attached to the surface. Then we also checked if the stretching force applied to GP1B receptors of the platelets is high enough to cause integrin activation. We measured it for different shear stress and also for different values of adhesion force parameters here. So basically these green symbols go for the wild type healthy platelets and healthy von Willebrand factor. This one goes for low adhesion case, and the red one is for high adhesion case. So we can see that the threshold of 10 piconewtons is breached for normal platelets at around 3 Pascal shear stress, while for the low adhesion case, well, it's not. It's almost not the case. Also, we compare the number of activated attached platelets for different kinds of model parameterization, and we see that for any case, after 1 Pascal, there is a rapid increase of activated attached platelets in the simulations. And also, we observe several platelets get activated, but they still keep flowing with the blood. So there are 3 platelets that are active, and they are potentially dangerous because they can provoke thrombosis in some other place along the flow. So with this model, we can also distinguish between healthy and von Willebrand disease cases. For instance, we can attribute these green lines that correspond to non-activatable von Willebrand factor, which means that both green and purple bits of this protein are attractive to platelets. We assign some potential energy for this attraction. And we see that for this type 2B von Willebrand factor diseased system, we observe a considerable amount of attached platelets for lower shear rate, lower than 1 or lower than normally 5 Pascals. And we see that there is still a big amount of activated attached platelets in the simulations in that case. To the contrast, if we consider type M von Willebrand disease protein, which is less sticky to platelets, we see low number of attached platelets, no matter what the shear stress is. And we also observe the low number of activated attached platelets in that case. Normally, we see that for normal parameterization in our model, the dependence of the number of both attached and attached activated platelets is nonlinear and non-monotonous. It has a maximal value at around 10 Pascals, and almost all attached platelets get activated in that case. And in any case, we consider the number of activated platelets that are present in the blood flow that are not attached to von Willebrand factor, that are moving freely, but they have active integrins. And this number, of course, grows with the increasing of the shear stress in the system. And to conclude, I would like to show several movies that were rendered from the numerical simulations. Here we can see the comparison between the adhesion of platelets with activatable integrins and with inhibited integrins for the shear stress of 4.5 pascals. And we see that in case of integrin activation allowed here, a greater number of platelets can adhere to von Willebrand factor. Next, there is a simulation with red blood cells, and we're focused on this kind of work right now. It's a simulation, a very small microvessel. It's almost a capillary. Interesting things happen here. We see that platelets are being pushed towards the vessel walls by those collisions with red blood cells, and eventually some of those platelets get a contact with von Willebrand factor, but when they get out here, they are being continuously pushed by red blood cells, and this pushing by collision with red blood cells also disrupts their adhesion to von Willebrand factor. Yeah, so not all platelets are able to get attached in such conditions, but apparently they experience very high tension forces that load their receptors, and probably those platelets get activated in this particular setup. So to conclude, I would like to line up several important points from this presentation. We developed a hybrid mesoscopic approach, and when I say we, I would like to address to our students, to our group, and even those who are alumni right now, who have graduated already. The hydrodynamic forces first, as we observe, unfold von Willebrand factor multimers, and after that they activate those monomers. Normally, the shear-induced platelet activation correlates with the activation of von Willebrand factor here. And also we see that platelets can get activated by mechanical forces, quite a feasible process, at least at the shear stress greater than 2.53 pascal. So elevated shear stress that could be observed in arterioles is basically one of the possibilities, one of the mechanisms that can activate mechanically platelets that adhere to von Willebrand factor. Thank you very much for your attention. I would like to conclude and give some acknowledgements to all the supporters, of course, the Moscow State University and the Russian Science Foundation.
Thank you very much, Alexey, for this really very, very interesting presentation. It's not the first time I am listening to you, and each time you add new and new elements to your model, it becomes really very, very complete and really very beautiful model. I look forward to see also the next step when you will introduce also blood coagulation in plasma with coagulation cascade. This will be even more interesting. But already now you presented so many different parts of your model and different factors acting together. And of course, there are many questions and comments. So please, if there are questions in the audience, you can ask them. Well, waiting for the questions from the audience, if there are any, maybe I will ask you something I was thinking about during your presentation. For example, about, well, what I also liked very much is this interaction between red blood cells and platelets. And of course, we discussed these questions, well, many times in different contexts. What can you say? Is there like biochemical or physical biochemical interaction between platelets and red blood cells? What is happening? Or just, first of all, in your model, it looks like it's pure mechanical, but maybe there are also some other types of interaction, right?
Yeah, thank you, Vitaly, for this question. Of course, it's a very complex thing to tell all the details, and probably I'm not aware of all the details of biochemics that takes place between red blood cells and platelets, but in this version of this model, they interact only mechanically. However, right now I work on introduction of the feature that allows for red blood cells to aggregate to each other. So it's known phenomenon of adhesion between membrane points of those cells, and the red blood cells can form those rouleaux formation, and it's very well-known phenomenon, and probably mechanically this has a great impact on what is happening in microvessels, and especially in stagnation point flow or aneurysms and so on, because as you know, this aggregation of red blood cells has a great impact on blood rheology in general. Secondly, we discussed earlier that there is a possibility that platelet contact with red blood cells in stagnant conditions may trigger blood coagulation in the vicinity of those conditions, but this really happens when the shear stress or shear rate in the blood is quite low. So it is mostly related to aneurysms, maybe left atrium appendage, probably, and well, stagnant areas when the flow is not very intensive. So in this particular work, we study the flow in arteries, and all the biochemistry is usually washed away from the place of the injury. And besides that, the biochemical reactions require a considerable amount of time, for example, several seconds or tens of seconds to give some significant result. And in this work, we focus on the very initial stages, like several tens or several hundreds of milliseconds after the exposure of von Willebrand factors. So it's not there yet, but I hope it will be implemented in some time in future.
And also platelet aggregation, you don't consider in your model here, no? Right?
They actually can bind to each other if there is a mediator. For example, von Willebrand factor in between those platelets, like in a sandwich, they can stick to each other. That's what it is. What is present currently.
You have it in the model, right?
Yeah. So two platelets can bind to one multimer, of course, and also there is such feature in the model that platelets can bind to each other through the integrins. The activated integrins can cause platelet sticking, but in this particular simulation, we just switch this off. So we set the parameter responsible for integrin binding to 0, not to blur the picture. So the integrins.
No, sorry, but in the left figure, it is written that there is integrin activation.
Yes, there is integrin activation, but we have different adhesion strength parameters for integrin binding to von Willebrand factor and integrins binding to other integrins, because this integrin binding is mediated by another protein, fibrinogen. So basically, we simulate the situation when the integrins can activate, but there is no fibrinogen in the model.
Yeah, okay. You also mentioned about positive feedback between platelet activation and von Willebrand factor activation, right? Could you please explain again? I think it's a very interesting point, so I think it would be interesting to learn a little bit in more detail what is happening here.
Yeah, what's happening here, when one platelet binds to von Willebrand factor, there may be not many activated von Willebrand factor monomers present at this stage in the system, but when this binding happens, this platelet is being dragged along the flow, and it causes the stretching of the whole multimer of von Willebrand factor, and several multimers that are attached to this platelet get stretched as well. And as we know, stretching of these von Willebrand factor multimers causes them to become more sticky to other platelets. So that's what I call the positive feedback loop. Maybe it's just too early to say that there's especially like feedback, or specifically maybe something different. Maybe it's just a pure mechanical event which has a less impact on the platelet aggregation at all. But we can see that, well, basically when one platelet binds, it provides the binding interface for other platelets. So,
yeah, I see. Well, in fact, yes, I understand what you say. I think I was a little bit mistaken. It's not platelet activation, platelets binding, which has this feedback, because in fact you don't consider here platelet activation, or you do consider.
It doesn't really matter, because the platelets can bind if the adhesion force strength is high enough to maintain this adhesion between platelet and von Willebrand factor for a considerable amount of time. Then this interface will be active for binding other platelets, and the activation may happen later.
Yes, but activation probably you can take into account platelet activation through integrins, right? But for example, their shape, of course, does not change.
Yeah, it's an important thing to mention that I missed in my presentation, that there are several degrees of platelet activation, and it's a quite new finding from experiments. And most likely there is an intermediate platelet activation degree when they maintain their spheroidal shape, but the integrins are already adhesive.
Yeah.
So usually what people say in the publications now, especially in this one that I referred to, is that the mechanical way of activating platelets causes mild calcium oscillations that only turn on those integrins, but they do not cause the shape changes, they do not cause the granular secretion, and so on. So biochemical activation of platelets may be a way to trigger platelets not completely. So this system is probably a sensor system, and the platelet is a doubtful unit here. It can bind through GP1B, but if the stimulus is not high enough, for example, well, there is not much von Willebrand factor, and maybe it's just a random kick was sensed by this GP1B, there's probably an inhibition mechanism inside the platelet that decreases the calcium level and then deactivates GP2B integrins.
Yeah, so main activator is calcium, right?
Main transmitter of the signal, I would say. So calcium provides inside-out signaling. It comes out from mitochondria, and oscillations of calcium trigger the integrins. But the stimulus can be different. For example, biochemical stimulus like ADP and so on. Those biochemical activation usually causes irreversible activation of platelets, but mechanics probably causes reversible, and that's intriguing. And not much is known about the distinctions, especially what happens inside the platelet, and it's still to be implemented in the model.
Another very interesting question you mentioned, and I think that some of our listeners paid attention to that, about possible relation between platelets adhesion, von Willebrand factor, and atherosclerosis initiation, because because this is one of the topics we discuss from time to time, and in particular Gada present here studies atherosclerosis development, initiation, things like that. We never, when we discuss these questions about atherosclerosis, we in fact never go in the detail how exactly monocyte adhesion happens to the endothelial cells. We remain at like more macroscopic level, that endothelial cells are activated by some inflammatory cytokines. They activate their receptors at their surface, and these receptors in some sense capture monocytes rolling vessel wall. But in fact, we never discussed the possible role of von Willebrand factor and platelets, but I think it's a very interesting question also. So we can think about that also. But behind atherosclerosis, of course, we know very well there is an inflammation. So from your point of view, are they independent of each other? I mean, platelets, von Willebrand factor, and inflammation, or they can somehow be related?
Well, this was a catch in my talk, this particular figure. I'm also very excited about this publication, and when I realized that the authors of this publication say that von Willebrand factor may be in particular involved in monocyte recruitment through the platelets, then it of course triggered my attention. What they say, it's a rather fresh publication, 2018, but there are some others which say pretty much similar things and address to pretty much similar mechanisms. So the activated platelets, which could be found in a decent amount along those von Willebrand factor strings here, they can expose P-selectin on their outer membrane after their activation. So there are some granules that turn out through the exocytosis, and they expose the P-selectin protein, and those monocytes have a receptor that specifically binds those P-selectin. So that's the mechanism that they report in this publication. Also, besides that, as I mentioned, those viable Platelet bodies exposed by the endothelial cells after inflammation on their membranes contain the P-selectin as well. So besides exposing von Willebrand factor that is meant to catch platelets, viable Platelet bodies and the inflamed endothelial cells also expose P-selectin that is adhesive to monocytes. Probably these initial stages and these two ways to expose the P-selectin meant to support each other. For example, those platelets protrude through the membrane, so platelets can, let's say, so they can catch monocytes at far distance, then pull them mechanically to the surface of damaged endothelium, and then monocytes can bind to this P-selectin on the membranes of endothelial cells. But just like an assumption maybe, and this should be verified. So I'm very excited about this experimental finding, and actually looking forward maybe simulating the molecular cellular processes, mechanical processes that take place at that level. So maybe we should discuss it somehow.
Yeah. So in this figure in the right, really very nice figure, this photograph. So the size here, it's 25 micrometers, so it's quite large, right? So in fact, as you say, if it goes inside the flow from the wall, inside the flow, so it can really then capture monocytes, because well, my understanding or our understanding was that monocytes just roll on the surface, but what you say, it can be more inside flow, right?
Well, maybe I'm mistaken, but I remember that there's a known mechanical effect that in the whole blood, the softer red blood cells concentrate in the center line of the vessel.
Yeah,
and
right.
Everything that is stiff is just being pushed towards the wall, including platelets and including white blood cells. And I think I remember there's a publication on a computational study that white blood cells with stiffer membrane are also pushed to the wall, and they roll along that wall. So
well, in fact, yes, in fact, we did such simulations already many years ago, yeah, where indeed we had red blood cells a little bit similar to what you showed, this deformable cells, and white blood cells which are kind of stiffer, like spheroids, and indeed they're pushed by these elastic cells, deformable cells, to the wall. Indeed, and this was really what we observed in the simulations, that these white blood cells roll on the surface because they are pushed out by red blood cells. But maybe not necessarily so. Yeah, but it's a good point that in fact this adhesion of monocytes to von Willebrand factor. So quite possible that all that, as usual, all that is so complex, and maybe they indeed somehow work together.
Yeah, what I'd like to add here, that it's obvious that these monocytes adhere where the platelets are present. And other areas probably covered with also p-selectin, but we cannot see good adhesion of monocytes here, but only where those platelets are present. Maybe just complex phenomenon of strengthening the adhesion in the place where von Willebrand factor is. So,
well, I don't know whether I can continue with my comments and questions if you are not already tired of that. But for example, you showed in one of the first slides that this molecules von Willebrand factor is not necessarily attached to the wall, but it can be like flowing with blood flow, right?
Yeah.
So I suppose that this is not occasional, and it can play also some physiological role. What it can be, platelet adhesion in the flow, or what is happening there?
Well, of course, von Willebrand factor is present in blood plasma in some concentrations. And normally, we actually know from simulations and also from some experimental studies that the free-flowing von Willebrand factor multimer is less susceptible to shear stress in sense that under the same hemodynamic conditions, the attached one is more stretched and more prone to activation than the free-flowing one. That's the first point to mention. Secondly, there is a phenomenon very interesting, which is known as self-association of von Willebrand factor. Whenever there is a inflamed endothelium and whenever it exposes ultra-large von Willebrand factor multimers from it, there is a possibility that this plasma-borne von Willebrand factor may aggregate with those strings to form longer strings. So probably what we see in that picture here is the result of this self-association. But important thing that the endothelial cells expose this ultra-large von Willebrand factor, which are much more activatable than normal von Willebrand factor, because they are longer, because they experience greater drag forces from the flow, and they can easily form those, thicker strings and bind more platelets. Therefore, we focus only on this part. But of course, it's possible to add the soluble von Willebrand factor.
Well, thank you very much for this very interesting presentation and also very interesting discussion. If there are no other questions, then I think that we will stop our discussion now. Thank you again, and you are very welcome to participate in our future seminars and to keep us informed about your ongoing work, which you built like that, brick by brick, during already many years. Okay, thanks a lot, and bye to everybody.
Thank you. Goodbye.
Thank you.
Thank you.
Thank you so much.
Thank you. Thank you and goodbye.